Today is part 2 of the story of using vaults as vaccines. I want to stress that this is a work in progress, and much of what I'm going to tell you about is still preliminary and subject to alternative interpretations. But that is the nature of research. Shortly after we learned how to package foreign proteins into vaults, I was giving a seminar about this to UCLA colleagues, and afterwards approached by Kathy Kelly, an immunologist who worked on chlamydia. Kathy asked me if we had ever considered using vaults as vaccines. As I mentioned last episode, I was just learning about immunology, and the only thing I knew about chlamydia was limited to a bad college joke, one that's probably best left in the 1970s. A teenager and says to her mom. The mom shrugs and says, "Your father will drink anything." If you like that joke, leave us a clap. Hi, I'm Lenny Rome, the Vault Guy. I'm a cell biologist and a nanotechnologist. My passion is a common particle of the human cell called a vault. With the help of my daughter-in-law, Tara, I am trying to explain what vaults are to non-scientists. As a bonus, I'm also teaching cell biology and some basic immunology. Today I want to cover 3 things. First, I want to review the sexually transmitted disease chlamydia and tell you what happened when a vault chlamydia vaccine was given to a mouse. Second, I'll describe the human innate and adaptive immune systems. And finally, I'll review the effects of adjuvants and describe why vaults might be a new type of vaccine. Stick around and find out if a vault vaccine can trigger an immune response. So, after Kathy asked me about using vaults as vaccines, she told me about her work on chlamydia and how efforts worldwide to develop a vaccine that protects from a chlamydia infection had been unsuccessful. Let's do a quick intro to chlamydia. Chlamydia is the most common sexually transmitted infection on the globe. It is caused by a bacteria called Chlamydia trachomatis. According to the World Health Organization, there are more than 100 million new cases every year. The infection often comes without symptoms, particularly in young, sexually active adults where prevalence is the highest and can lead to serious health issues if left untreated. Kathy's goal was to create a vaccine that could protect humans from chlamydia. This was very difficult for many reasons. For starters, chlamydia is largely caused by an overreaction of the infected person's own immune response, causing an inflammation. Traditional vaccines that trigger the production of antibodies don't work against chlamydia, since chlamydia does not appear to be prevented by antibodies. Instead, prevention requires the production of specialized immune cells. The human immune system has developed 2 ways to respond to foreign pathogens: a general and a specific system. The general system, also called innate immunity, is something you get at birth and has several lines of defense that are always active. This includes barriers like skin and mucous membranes and certain natural or innate immune cells that recognize most pathogens. These cells respond quickly and generally to any threat. They are not specific. In contrast, the adaptive immune system is slower to respond, but highly specific. It learns from each infection. This response takes longer because specialized immune cells and antibodies need to be produced. Adaptive immunity can last a few weeks or months. In some cases, it can last for the rest of a person's life. To make things more complicated, this is the immune system we're talking about after all. There are 2 forms of adaptive responses. They are called humoral immunity and cell-mediated immunity. The word humoral comes from the Latin for liquid or fluid. Humoral immunity uses antibodies which are proteins that fight pathogens, like bacteria and viruses in the body's fluids, which used to be called humors. Both forms of adaptive immunity, humoral and cell-mediated, rely on specialized white blood cells, B cells and T cells. B cells produce antibodies, while T cells are the key players in cell-mediated immunity. Kathy thought that perhaps we could target vaults to cells of the immune system that can stimulate both forms of adaptive immunity. I thought this was an interesting idea, but I explained to her that empty vaults had no effect on the immune system, so we should start with a simple control experiment where we just package a chlamydia antigen into a vault and attempt to use it as a vaccine. Kathy's lab had a system set up to test vaccines for their ability to protect a mouse from a chlamydia infection. Mice can handle chlamydia much better than humans because after one chlamydia infection, they develop immunity. We made empty vaults and packaged them with a chlamydia antigen that had been previously shown to trigger the production of antibodies in mice. The antigen we selected was a protein found on the chlamydia cell membrane called major outer membrane protein, abbreviated MOMP or MOMP. We created the MOMP vault by expressing the MOMP-int fusion protein and packaging it into the empty vaults, a process I described in episode 14. We started with MOMP because we wanted to be sure that we used an antigen that was known to induce an immune response. This is often referred to in science as a positive control. At this point, we just wanted to know if vaults containing an antigen could stimulate an immune response. We had 3 groups of mice. The first group was exposed to a live chlamydia bacterial infection. We knew that after 2 weeks, this group would clear up their infection and become immune to a second infection. The second group of mice was immunized with the MOMP vault. We also immunized a third group of mice with a vault containing an antigen that was not found in chlamydia. This group served as a negative control. After waiting a few weeks, all 3 groups of mice were then exposed to live chlamydia. In vaccine talk, this is called a challenge infection. The results were very interesting and at that time completely unexpected. First, the mice that were previously exposed to chlamydia were now immune. This was what we expected. Second, when we looked at the mice vaccinated with the control vault, remember, this vault was packaged with a protein unrelated to chlamydia. Also, as expected, there was no protection. These mice developed an infection similar to what they would have gotten had they not been vaccinated at all. Now, here is the unexpected result. To our surprise, we found that the mice vaccinated with the MOMP vault were protected from the chlamydia challenge. This protection was as equal to or better than the protection seen in a mouse that had recovered from a previous chlamydia infection. We were really blown away by this data. We didn't understand how a vaccine composed only of a vault packaged with a chlamydia antigen could protect the mouse. Remember, we did not add an adjuvant and we did not target the vault to the immune system. When we examined the mechanism of protection imparted by the MOMP vault, we found that there was a stimulation of both forms of adaptive immunity. We saw the production of anti-MOMP antibodies and the production of beneficial T cells that could reduce inflammation. This was completely unexpected. Wow, we realized that this was not going to be easy and we were going to have to go back to basics and examine how the vaults could be doing this. I'm Lenny Rome, the Vault Guy. Join me in the next episode where I describe experiments that shed some light on the seemingly remarkable vaccine properties of the vault. Please remember to like and subscribe. I really appreciate the small group of vaulters that watch every episode. You guys are awesome.